Hello and welcome. In this tutorial, we will discuss about cell, tissues, organ, and organ system of the animals, and basic organization of zoology. For chemistry, we have to understand atoms, and for the zoology, because all living organisms are made up of cells. Cell is the basic unit of structure and function in living organisms. So we have to study about cells and different tissues and organ system levels. And if we look around the cells, what are the cells? It is the basic unit of structure and function in living organisms. And types of cells are basically two types of cells: prokaryotic cells and eukaryotic cells. Prokaryotic cells are the basic type of cells, and we can also divide the prokaryotic and eukaryotic cell into two main domains, like archaea have the ancient type of organisms, and the prokaryotes means pro means before, and eukaryotes being nucleus, they have no definite nucleus. And eukaryotes are membrane bound nucleus, and they have a nucleus and a different type of membrane bound organelles, like mitochondria, cytoplasm, endoplasmic reticulum, that are not evenly present in prokaryotes. And eukaryote cells basic structure is made up of plasma membrane, outer membrane boundary of the cells which separate the cell from another cells, are from the extracellular material. It is the plasma membranes. And plasma membrane also has the specific receptors for the molecule that alter the cell functions, and for play important role for signaling. We will learn about it in detail in next chapter. Then there is the cytoplasm between cell nucleus and cellular membrane, in which organelles are present, and in the center we have a nucleus. This is the particular type of the structure of the cells we can understand. Like if we open this particular type of the cells, we will understand what type of the organelles and what type of structures are here in the cells. It is consists of outermost boundary called cell membranes, and it is also called present a nucleus, which is in the center, which is the station of control. And there is the cytoplasm in between nucleus and cell membrane. It contains various organelles, like this is mitochondria, nuclear envelope, nucleolus, chromatin, Golgi apparatus, vesicles, free ribosomes, endoplasmic reticulum, rough endoplasmic reticulum, lysosomes, Golgi apparatus, mitochondria, and every kind of cells. Cells are basically very small in the measurement of micrometers. What are the basic reason why cells are small? And that cells should have are having small nucleus. Cell have small nucleus, and nucleus size of nucleus to size of cytoplasm should not be a very much great, and it will be difficult for controlling the nucleus to enter cells. Like all the activities are controlled by nucleus, which contain DNA, and it form mRNA, messenger RNA, and form proteins, and it help control on all the organelles of cells. If the nucleus is small and cell become very large, it will be difficult for nucleus to control the activities. And second is that cell surface area to volume ratio should be great, and when the cell size increases, its volume increases more markedly than that of the surface area, and it will be difficult for exchanging of the material from different type of cells, to exchange material between between different type of cells, and between extracellular matrix, and between out of the cells. So it should be have a small volume and great surface area for proper functionings. And this is the second reason why cells are the very small. Like you can understand that this is our surface area, this is the our radius. Cells, if the radius of the ball is 1 centimeter, its surface area is 12.4 0.57 or its volume is 4 and surface area to volume ratio is 3, then we double the radius and surface area also almost full time become full time, but volume increases to as many as about 16 times, and surface area to volume ratio decreases to 1.5, then it will doubled further its surface area to volume ratio decreases further. So increasing the cell size, surface area to volume ratio decreases, and it is difficult for them to control of the nucleus for the cells. And we will discuss about the basic organization of the cells, like simple to complex ones, like living organisms are made up of atoms combined together to form simple molecules, then they will form macromolecule, then will form membrane, then organelles are combined together to form the cells. A cell is the basic unit of structure and function in living organism. All these things, organelles, membrane, micromolecules, are independently non-living organisms. Then the cell combine to form tissues combine to form organ combine to form organ system, and organ system will develop animal. Then we will also study about populations, community, this type. Here we will discuss about difference between prokaryotic and eukaryotic cells. That is also very important, like membrane bound nucleus is absent in prokaryotes but present in eukaryotes. DNA complex with the histone is not present in histone prokaryotes but present in eukaryotes. Number of chromosomes is 1 in prokaryotes, and eukaryotes more than 1 or maybe 1. And nucleolus absent, there is no nucleus, there will be no nucleolus, and nucleolus is present inside the nucleus. Mitosis occur in prokaryote. No mitosis occur in prokaryote, but mitosis occur in eukaryotes. Genetic recombinations, that partial unidirectional transfer of the DNA. Then meiosis and fusion of the gametes is the genetic recombination in eukaryotes, and it is unidirectional transfer of the DNA, like DNA from RNA from protein inside the cytoplasms. Mitochondria are absent in prokaryotes and present in eukaryotes. Chloroplast absent in prokaryotes and present in eukaryotes. Plasma cell membrane usually no and present in eukaryotes. Flagella are the submicroscopic in size, composed of the only one fiber for prokaryotes, and flagella of eukaryotes is microscopic, can be visible with the light microscope, and usually about 20 microtubule 9 plus 2 pattern is found, 2 tubules in center and 9 around these two. This is the specific patterns, like and protein and flagella in prokaryotes may be flagellan proteins, but it is microtubule and tubulin proteins in eukaryotes. Endoplasmic reticulum is absent in prokaryote, present in eukaryote. Golgi apparatus absent and present in eukaryotes. Cell wall usually chemically more complex. Most important feature of the prokaryotes is that they have peptidoglycans and murines and very complex cell wall, succulus materials, chemically simpler in eukaryotes. The other organelles like ribosome also smaller in prokaryote, 30s ribosomes and 80s ribosome in eukaryotes, but mitochondrial and chloroplast are all like prokaryotic, and these are of 70s. Swedberg unit for ultracentrifugation. Lysosome and peroxisome are absent in prokaryote but present in eukaryote. Microtubule are absent or rarely present, but they are present in eukaryotes. Cytoskeletons may be absent but always present in eukaryotes. Vacuoles present, vesicles present in both type of prokaryotes and eukaryote. Differentiations may be rudimentary, and there are no differentiation of the cells to tissues and organ systems, and in eukaryotes there is the formation of tissues, organ systems. We will discuss later on.